Hi, my name is Parag Meswani, and I'm the Chief Commercial Officer for Sio Gene Therapies. It's my pleasure today to present on behalf of my many colleagues here to discuss the company and our programs. But first, I want to thank the meeting organizers for giving us the opportunity to present here today. These are our forward-looking statements, and as a reminder, I may be making forward-looking statements as part of this presentation. So I joined Sio Gene Therapies about 2 years ago from Spark Therapeutics, and we have been on this journey to build an amazing company and to develop gene therapies that have the potential to radically improve the lives of patients. And we intend to do that across the 3 pillars that drive our approach and that drive our strategy. These are platform, partnerships, and of course, people. First, with platform, our goal at Sio Gene Therapies with our programs is to ensure that we have the right product and indication fit. That means that we try to leverage the right vector systems with the right routes of administration to get the gene therapy where it needs to be. And in effect, we try to have a balanced approach to how we do drug development, where we're using both AAV and lentiviral vector systems for our programs. We're using both direct to CNS and systemic delivery methods to ensure that that product indication fit is sound. And we believe that we have the potential to develop and deliver first-in-class and best-in-class gene therapies across our entire portfolio. And indeed, we're seeing emerging clinical data in 2 of our indications, in Gm1 gangliosidosis and Parkinson's disease, that supports this. Second is partnerships. We have programs that are built on a foundation of science and research from leading gene therapy organizations across the country and across the globe. These include groups like the University of Massachusetts and Auburn University, who have done seminal research for our rare disease programs, to the NIH, who is one of our premier clinical trial sites for our Gm1 program. We're partnered with top AAV and lentiviral manufacturers like Viralgen, who is an AskBio subsidiary, and Oxford Biomedica, who has pioneered some of the work in the lentiviral manufacturing space. And we supplement and support that with our own efforts on preclinical and analytical development in our new North Carolina laboratory facility. But last but certainly not least, we have a foundation of excellence in the people that we have attracted here to Sio Gene Therapies, who not only have deep CNS and gene therapy expertise, but who share my commitment in being a patient advocate for the rare disease and Parkinson's community that we intend to serve with our programs. And so this is a snapshot of our clinical stage portfolio of gene therapy programs. As you can see, we've designed a pipeline that we believe has balance, where we have very classical applications of gene therapy using AAV-based approaches for our programs in Gm1 and Gm2 gangliosidosis, respectively. And we have our Parkinson's program that, in effect, delivers 3 transgenes using the larger packaging capacity of the lentiviral vector system to deliver benefit. All 3 of these programs are in the clinic, and we look forward to a number of different milestones throughout this year and into next that I'll articulate in this presentation to support the overall product profile for each of these indications, which we own worldwide rights for. First, let me start off with our programs in rare fatal pediatric diseases in Gm1 and Gm2 gangliosidosis. As I mentioned at the outset, we believe that these are very classical applications of gene replacement. For both disease states, these are universally fatal. They're both pediatric lysosomal storage diseases. And importantly for gene therapy, these are both monogenic autosomal recessive diseases where we know what the disease-causing mutations are. In the setting of Gm1, this is caused by mutations in the GLB1 gene. And the consequence of that is an impaired production of the beta-gal enzyme and the accumulation of Gm1 ganglioside in multiple organ systems in the brain and in the periphery that leads to significant deficits. In the setting of our Tay-Sachs and Sanoff program, these are due to mutations in 2 genes, in the HEX-A and in the HEX-B gene, both of which are requisite to create the beta-hexa enzyme, which is deficient in both Tay-Sachs and Sanoff disease, And our goal here is to deliver both of these transgenes to treat Tay-Sachs and Sandhoff patients. In both of these indications, there are some unique features that make them really attractive targets for gene replacement. So if you look at this slide here, this is the biochemical pathway where you see that both Gm1 and Gm2 gangliosidosis are part of a singular continuum. And in both indications, there is an underlying neurobiology that supports the use of gene therapy for long-term enzyme restoration. Both indications are able to leverage this notion of cross-correction, where a little bit of enzyme can go a long way in changing disease phenotype, or we can achieve broad distribution via single administration. And finally, both indications benefit from having really well understood understanding of the natural history of the diseases. And this has informed how we design our clinical trials and how we aim to understand whether these gene therapy programs are providing true, meaningful benefit for the patients that need them. And before I get into the programs in a great amount of detail, I wanted to remind the audience that our program partnerships are built with leading global gene therapy institutions, where we have begun to already lay the foundation for commercialization through manufacturing partnerships with Andelin Biosciences, which is a premier manufacturer of AAV technology formed by Nationwide Children's Hospital. And we're beginning the commercial scale-up process for our programs, partnering with Virogen, which is a leading CDMO built as a joint venture between AskBio and Columbus Venture Partners. So first, let me start off with our Gm1 program, which we believe is a best-in-class gene therapy for the treatment of Gm1 gangliosidosis. And it's built on a strong scientific rationale that builds on this principle of product indication fit. Here, we're using the proven AAV9 vector system to deliver a functional copy of the deficient GLB1 gene through IV administration. And this is a fundamental differentiating feature of this program, where we believe that delivering this gene therapy through a systemic or IV route of administration is requisite. It's needed to address the CNS and peripheral manifestations of this disease. And indeed, in naturally occurring models of disease, this is the only gene therapy to demonstrate restoration of wild-type survival. And that's an important differentiator for this program that's not been demonstrated previously. You can see some of the data here supporting this thesis, where in an independent study conducted by Auburn University, comparing our Gm1 gene therapy approach, this is the AAV9 IV approach, with two other Gm1 gene therapy programs, you see in very stark terms the ability to have this route of administration and this vector have superior CNS distribution in a Gm1 feline naturally occurring model. What you see here on this slide are cross-sections of the brain and the spinal cord. The deeper the staining, the greater the expression of the deficient enzyme, and the more normal you look like. And you see in very clear patterns the differences between the three distinct Gm1 gene therapy programs. This is using the very same vector, the very same dose that we've now taken into the clinic. And so this is a summary of our clinical study design in our program in its totality, where we're starting in a dose ranging phase with a low dose of 1.5 E13 and moving on into a high dose cohort of 4.5 E13. And as we define the safe and efficacious dose, we'll move into stage 2, which is an efficacy-focused part of the program. We are looking at a host of measures, inclusive of safety, biomarker-based activity, looking at enzyme activity production, along with measures of development. And indeed, we've already shared data at 6 months, just this past December, in 5 children that were part of our low-dose cohort. And what this particular readout showed us, and this is just one example of the data, is the ability to produce enzyme and to produce it reproducibly starting at the earliest measurable time point 1 week after gene transfer and being sustained out to the day 180 observation period. This in effect demonstrates a rough doubling in enzyme activity in the serum across 9 distinct time points. This is supported by additional evidence that showed the potential for disease stabilization in a disease state where we know there is a very predictable age-related decline in functionality. So we're really encouraged by the data we've seen at the 6-month mark, and based on that, we've already moved on to a higher dose cohort. In parallel, we've begun foundational commercialization and launch preparation activities. These include continued engagement with patient groups and with physicians who treat children with Gm1 gangliosidosis, I mentioned already the manufacturing approach that we've taken and the partnerships we've already begun to build and execute upon with ViralGen. And we've begun to lay the foundation for commercialization to enable a faster diagnosis and the potential to treat these children as early as possible in the disease continuum to give them the best chance at having a meaningful outcome. And so where we are right now with the Gm1 program is we've shared positive clinical data where we've shown the ability to have an increase in enzyme activity and preliminary evidence of disease stabilization in late infantile and juvenile children treated in our low dose. Based on the outcomes observed there and the safety generated thus far, we've now moved on to a higher dose cohort of 4.5E13, and in total, 7 children have been treated as of February of this year. So as we look ahead, there are a number of different milestones to keep an eye on. In the second half of 2021, we're expecting 12-month data from our low-dose cohort. In the first quarter of 2022, we're expecting 12-month data from the first 2 children treated in the high-dose cohort. And then later in 2022, we're expecting an expression of durability, where we look to have 24-month data from the low-dose cohort. And all of these will collectively support a conversation we intend to have with regulators to discuss the path to registration and ultimately to approval. So quite a bit of progress to be made, quite a bit to look ahead towards for the remainder of this year and into the next for this exciting Gm1 gene therapy program. Let me move on briefly to our Gm2 program. This is AXO-AAV-Gm2, our gene therapy candidate for Tay-Sachs and Sandoff children. The thesis and the rationale here is that one-time delivery of 2 vectors using the AAV-RH8 vector system that encode for the HEX-A and HEX-B genes has the potential to increase the deficient enzyme, in this case, the beta-HEX-A enzyme. And our goal here is to deliver these gene therapy vectors directly to the CNS using a very unique dual route of administration, both into the thalamus and into the spinal canal, to allow for transduction across the entire neuroaxis. This same approach has been tested in naturally occurring Sandoff models of disease and has shown the ability to have an increase in enzyme activity and an extension in survival. Based on this, we've now taken this program into the clinic, where we received IND clearance back in November of 2020. And just this past January, we announced that we dosed the first patient in our AXO-AAV-Gm2 clinical trial, along with our collaborative partners at Mass General Hospital and at the University of Massachusetts. So far, the therapy has been generally well tolerated to date with no serious adverse events. So overall, very encouraging from what we've observed so far. And the next steps are to continue enrollment in our dose escalation series known as stage 1. And you can see a summary of our clinical program here on this slide, where we start in stage 1 across a low, medium, and high dose of AXO-AAV-Gm2. And once we identify that safe and efficacious dose, our goal is to move into a stage 2 efficacy-focused study. This is the totality of our clinical program, where we intend to treat 16 to 20 children all in between stage 1 and stage 2. And our goal is to assess the safety of the gene therapy, the safety of the delivery mechanism, and ultimately define whether this therapy is efficacious across a host of different clinical measures. These include developmental milestones, motor function, and disease severity, biomarkers, including enzyme activity in the CSF and in the serum, as well as Gm2 ganglioside levels in the CSF, among a host of other exploratory measures, including systemic and CSF biomarkers and an assessment of neurologic symptoms. So that next step will be to continue to identify patients, screen them, and enroll eligible candidates into stage 1 of the study throughout this calendar year. Now let me move on to our program in Parkinson's disease. As you guys may be well aware, Parkinson's disease is a progressive and chronic condition impacting 1 million patients here in the US and many million outside of the US. The challenge with Parkinson's disease is this notion of balancing motor function improvement with treatment emergent side effects. The gold standard, levodopa, is now 50 years old. And while effective in early disease, with disease progression, you have an increasing pill burden, you have a worsening side effect profile, a worsening impact on dyskinesias, and the benefit you derived earlier in the disease just isn't there anymore. And so there's a substantial opportunity to have a treatment that can improve motor function and quality of life without treatment emergent side effects like dyskinesia. That's where AXL NTPD comes into play, where this is a one-time treatment using a lentiviral vector system that delivers the three key enzymes involved in the endogenous production of dopamine. We've generated data across 21 patients in 5 distinct dose cohorts that have shown dose-dependent improvements in motor function, increases in quality of life without those dyskinesias that have held back treatments in the past. And the next steps are to continue to scale up our manufacturing process using a suspension-based approach that'll deliver the requisite clinical trial material to advance our program into the next step. And as a reminder of AXL NTPD and how it works, as I mentioned before, here we're delivering the 3 enzymes required for the endogenous production of dopamine. These are TH and CH1 that convert tyrosine into levodopa, and AADC that converts levodopa into dopamine. And our goal here is to create steady, tonic, predictable levels of dopamine. And we can do this and we can deliver all 3 of these transgenes by leveraging the larger gene packaging capacity of the lentiviral vector system that is delivered direct to the target tissue in the putamen. What we've seen so far is a totality of data across cohorts that suggests a very consistent patient benefit, a very well-tolerated safety profile. We've generated data showing improvements in motor function on gold standard scales like the UPDRS off score at 6 months. We've demonstrated the ability to improve dyskinesias as measured by the Hauser diary. But most importantly, from a patient perspective, we've generated data that shows that AXL NTPD can improve the lives of people living with Parkinson's disease, and that's what this program is all about. One illustration that we think speaks to the power and the potential of AXL NTPD is illustrated here, where we're looking at the change from baseline on the UPDRS Part 3 assessment of motor function. And what we know, and you can see this illustrated in the yellow line, is that these patients will continue to worsen over time. With progressive disease, motor function will worsen. What we've seen so far across 5 different dose cohorts, which you can see in gray for PRO7, which is a first generation construct, in the aqua and teal color, you see the AXL NTPD data. And the goal here is to essentially turn back the clock, to give these patients a chance at having an improved quality of life and an improved motor function that is sustained beyond what you might have expected on standard of care and based on expected symptom worsening over time. This is the power and the promise and the potential of AXL NTPD illustrated on a single slide. And so what are the next steps for the clinical development plan? The next steps are for us to develop a reliable suspension-based manufacturing process. This is already underway with our colleagues and collaborators at Oxford Biomedica, who have initiated the manufacture of several GMP batches using the suspension-based manufacturing process. And once that is available, our goal is to file an IND here in the US and to file an IMPD in Europe to continue our clinical development plan, to continue to define the safe and efficacious dose, and ultimately enter into a pivotal clinical trial that will provide us with further guidance on the overall product profile for AXL NTPD. So let me summarize here with this slide, which really encapsulates the promise of the company and the work that myself and our colleagues are focused on for this year. First and foremost, we are laser focused on our 3 clinical stage programs, where we have a balanced pipeline that leverages proven technologies and approaches with both AAV and in vivo lentiviral vector systems and programs. We have very classical and some might say de-risked applications of AAV-based gene therapy in monogenic diseases, in Gm1 gangliosis, Tay-Sachs, and Sandhoff, coupled with an extension of the potential of gene therapy into more prevalent indications and conditions like Parkinson's disease. All these programs are in the clinic. All of them will be generating data. All of them have the potential to be best in class and first in class. And we're going to drive and leverage our broad gene therapy expertise and the team that we have here to drive these programs forward and to do our part to make an impact in the lives of patients and caregivers impacted by Parkinson's, Gm1, and Gm2 gangliosis. We have the means to do it with a strong balance sheet. We have the team to do it, and we have the platform to deliver on this promise. So quite a bit to look ahead towards for this year and into next. I want to thank you again for your attention and for your time, and I want to thank the meeting organizers.